Hi everyone, my name is Kyla LaBianca, and today I'll be talking about this unique and somewhat odd microbacteriophage named PineapplePizza. This phage is a podoviridae and it has a non-contractile tail and a pretty short genome of only 23 genes. It's also classified as a singleton phage, meaning that it's not similar enough to any other microbacteriophage to be included in a cluster. But that's not the only thing that makes this phage weird, and I'll explain what I mean later. In order to annotate this genome, I navigated to PineapplePizza's Pecan webpage to see all of the existing information on the individual genes, including suggested functions, blast hits, and HH-pred hits, among other information. I also used Famirator to make fan maps with other genomes for comparison. You might be wondering how I did this with the singleton phage, but I just took other phages from how they appeared in blast hits and HH-pred hits. So now for the weird part. While I was going along annotating these genes, I noticed that a certain other phage was popping up a lot in the blast and HH-pred hits. And it turns out that this phage is really similar to the genome of Bacillus subtilis phage 529. And this phage was among the very first bacteriophages ever discovered. And it was first found in 1965. It's old. So both phages are podoviridae and relatively similar in length, about 16,000 base pairs for PineapplePizza and 19,000 for 529. Although PineapplePizza is a little more GC rich than 529, as you see in my results, the number of genes are actually pretty similar. So PineapplePizza has 23, 529 has 27. If you look at the electron micrograph images here of both phages, you'll notice that they do appear pretty similar in structure, with the short-tailed and little arms coming off the sides. Those are appendages, which I'll get to later. Since PineapplePizza was newly discovered, there's not yet a higher quality projection of it like the 529 one on the bottom, but the picture on the right shows a simple representation of 529 with the genes and the numbers that correspond to different structural functions. So we have the head at gene 8, the tail knob at gene 9, the connector at gene 10, the lower collar at gene 11, and the appendage at gene 12. So it turns out that all 5 of these genes actually have synteny and share the same exact function as genes 8 through 12 in PineapplePizza, even though some of the function names in the picture are a little outdated and we use different names today. So remember that 529 was discovered in 1965, so of course we have outdated names. So if you look at the fan map on the bottom there, you'll see the updated names and that the functions are in fact the same. So gene 8 is a capsid, which is the same thing as the head. Gene 9 is the tail, same thing as the tail knob. Gene 10 is the portal, which is the new name for connector. And gene 12 is a tail spike, which is the updated name for an appendage. For gene 11, however, the function of lower collar protein is not registered as a C phage's official function, yet almost all the evidence pointed to it as the function for that gene. So to my knowledge, it's currently being looked at further, and there might be a different name for it later, but for now we're going to stick with lower collar. Another interesting thing I found is that when PineapplePizza was discovered here at UMass, it was suspected that both ends of its genome were linked to a terminal protein. And while this hasn't been confirmed, it is confirmed that the ends of PID29 are linked to a terminal protein. So the similarity between these two phages definitely suggests that PineapplePizza is bound to a terminal protein. So I just told you all about the similarity between these two phages. The weird thing about this is that these phages infect different hosts. So while yes, they probably would have some genes with similar functions, it isn't expected that they would be in the same exact place in the genome. So the big question is, why are these phages so similar? In order to answer this, it might be helpful to consider if PineapplePizza is similar to any other genome, and in order to do this, it might be helpful to get a more detailed electron microscopy projection of PineapplePizza. It also would be useful to experimentally verify that PineapplePizza does in fact have a terminal protein, because that was a part of the similarity. So that's my research. I'd like to thank my 2 professors, Dr. Jessica Rochelow and Dr. Peter Chen, for helping me along the way. Thank you.